Welcome back. I hope you've all had a snack. We've got a great lineup of speakers. To open the next session on the portfolio of tools and technologies to enable low emissions farming in New Zealand, I'd like to welcome Naomi Parker. She's the Executive Director of the Ag Emissions Centre and hopefully a familiar face to most of you.
Kia ora koutou katoa. It's my pleasure to provide the opening talk for our technologies update session, providing a bit of an overview of the Ag Emissions Centre portfolio. So just firstly, a very quick update on who we are. So our vision is healthy environment, thriving farmers, and delivering on New Zealand's agricultural emissions commitments, whether those are regulatory or market-driven, with a mission to accelerate evidence-led solutions for low emissions farming. We were launched in 2010 as the New Zealand Agricultural Greenhouse Gas Research Centre and rebranded last year as the Ag Emissions Centre. We're funded by government and sit alongside AgriZeroNZ and the Ministry for Primary Industries as partners in accelerating new mitigations. For over 15 years, we've grown in our funding and in our capabilities. What we've maintained is our focus on developing solutions for farmers, funding high impact research, building and investing in greenhouse gas measurement infrastructure, and helping to grow the incredible capability that contributes to this work across New Zealand. Our strategic objectives drive and shape our work. Cracking the fundamentals is the critical fundamental research that helps us understand agricultural emissions and its mitigation. Driving for proof of concept is what it says on the box, going from an idea to a tool or technology that has real world promise. Accelerating low emission solutions is the work that we support once a technology is proven to look at how to make it grow faster. And making low emissions farming a reality is all about how emissions reduction technologies and other actions add up on farm to reduce emissions. And we have two underpinning objectives: strong people capability, robust greenhouse gas measurement infrastructure, and clear communication in our enabling environment. And across all of our objectives is our focus on maintaining New Zealand's global leadership in this area. This is what those objectives look like in practice. Having a strong portfolio is absolutely critical. We're now using the technology readiness levels approach developed by NASA in the 60s for the moon landing program to map all of our R&D projects, and you saw this earlier in Hayden's presentation. Clearly capturing where these are up to and where we want to get them. 9 clear steps from an idea to an available technology. Each white box represents a project or program, and the lines to the right of the boxes are where we expect them to be in 2030. There's a lot on this slide, so I'm just going to focus on a few areas that cover the breadth of our objectives. Our key Cracking the Fundamentals project is the Chief Trial, and Sinead Leahy, the last speaker in this session, will be talking about that, so you should look forward to that one. In driving for proof of concept, we have an important focus on feeding the innovation pipeline. These are initially smaller projects aimed at getting to an experimental proof of concept. The 5 boxes that you can see there in a cluster represent projects that have been supported following our last 2 contestable innovation rounds. These are in the early stages, so we look forward to updating you on those in the future. And we're currently evaluating another round of that. And a whole range of research organisations, both domestically and internationally, are involved in those, and we also co-fund some of those with AgriZeroNZ. The End-of-Fight Delivery Project is a 3-year program including researchers from Victoria and Canterbury Universities, the Bioeconomy Science Institute and Grasslanz Technology. This stretchy project is investigating whether inhibitor compounds can be biosynthesised within epicloe endophytes, naturally occurring fungal partners of perennial ryegrass. If successful, this could create a new pathway for delivering mitigation through pasture renewal. We also have a really strong focus on low methane genetics. We know you can breed for low emissions genetics, and our investments are about pushing that forward. We've had a big focus on low methane dairy, and Richard Spelman from LIC will be covering off the progress on low methane for dairy shortly, including the challenges of that. We've now also kicked off a beef program, working with the Bioeconomy Science Institute and Beef and Lamb. To make all genetics work go faster for research and commercially, we know we need large numbers of measurements. So we're investing in a proxy program led by the Bioeconomy Science Institute, trying to identify cheaper and faster measurement approaches. This is also in its second phase, following some good progress in the first project. This could lead to a much simpler approach, such as a milk test or a buccal swab. And while the sheep work is already in a commercial environment, we're continuing to invest in this area, including jointly with MPI, AgriZeroNZ and Beef and Lamb, on further testing of rams and checking the relationship between feed efficiency and low methane genetics. These projects have an aim of increasing farmer confidence to adopt, and I'll touch on that further in a moment. And finally, we've been running a future farm systems research program since 2022. This work, undertaken by AgFirst and Parent Ag, is supporting collectives of farmers to explore low emissions farming, and also looking at the potential role of more transformational land use change. A white paper released last year summarises some of the key insights to date, which are strongly supported by other analysis and research that we've done. Some of those key insights include that change is possible at a farm level, but that there are limitations in terms of scale and in the commercial realities of farming. That it can be easier to explore and undertake the journey to low emissions farming with others rather than as individual businesses, and that farmer confidence in mitigation technologies is affected by a whole range of considerations, including market acceptance, cost or practicality of use, and the need for incentives to stack up. And we're working through shaping up what our farm systems work needs to look like in the next phase. Which is a good segue to the final area I want to touch on. Understanding, tracking and driving different mitigation approaches from a technical level is only one part of the story. We all know that if farmers don't use these technologies, then we've failed in what we're trying to achieve. Over the last year, we've ramped up our focus on adoption. Alongside the TRL approach, we've also added an adoption readiness analysis. This was developed by the Department of Energy in the USA a few years ago. This uses a series of questions about value proposition, market acceptance, resource maturity, and licence to operate to analyse adoptability. Delving into this analysis helps us identify where we and others might need to invest to increase adoption, from the earliest stages of research right through to accelerating on-farm uptake. The feed conversion efficiency trial for sheep is a great example of addressing a confidence gap. We then bring these together to map our investments with the aim of getting high-impact tools and approaches into the top right-hand box, highly technical ready and highly adoptable. So just to finish, some key insights from looking at our portfolio and our progress. Firstly, a broad portfolio. This is key to increasing likelihood of success, but also critical to giving farmers options that will work in their systems. Secondly, we need to continue to feed the pipeline and take calculated risks. Not every technology will ultimately get across the line, and in fact, if all projects succeed, we probably haven't got our risk settings right. As well as driving new tools, we need to keep looking at the fundamental science of emissions. Continuing to invest in people and infrastructure is key. The PhDs and postdocs we've funded over many years are now making critical contributions to research, and the infrastructure we invest in is supporting projects across the country. As I've just touched on, we need to be focused on adoptability at every stage of research. And to sum up, we need to keep looking at our portfolio, leveraging our networks, insights and data to ensure that what we invest in has the best chance of delivering real impact for New Zealand farmers. Thank you.
Thanks, Naomi. It's really lovely for me to see how we have moved from talking about inventing things and explaining the 101s of what we're inventing to so much focus on adoptability now. Now, our next speaker is Wayne McNee. He's the Chief Executive of AgriZeroNZ. He's going to talk about acceleration and pace and advancing technologies and timelines to get into the market.
Kia ora tātou, ko Wayne McNee taka wingwa. I'm Wayne McNee, Chief Executive of AgriZeroNZ. If you were here yesterday, I understand you probably already know who we are. We may have been mentioned occasionally already. I'd just like to also acknowledge Naomi and her team who've done a huge amount of work setting this conference up, and members of my team who are also here who've helped support that work. So really do appreciate it. It's been a great conference so far. Also noting there's been a lot of discussion about how important it is, and it is important. The farm systems change that is already happening on farm efficiency, and the contribution that makes to emissions reduction, or reductions at least in emissions intensity, is continuing and is very important. There are many tools already available, but what I'm going to talk about mostly are the tools that are still in development. We're focused mostly on those additional tools, and also wanted to acknowledge our farmer focus group, many of whom are here today, and appreciate them coming along to the conference. So we are a private-public partnership, and it says there what we're focused on. I'd really like to thank the private sector investors as well as the government for investing in us and having confidence in us. We set up in February 2023. It was a big leap of faith at the start for the private sector and the government to put funding in. So far, 191 million of funding has been contributed, and recognise that those investors who put their hard money down and have worked closely with us alongside the government to help set us up and get us underway. We work across a range of different areas. We have a catalyst team that focus, remembering actually when I say team, we're a total of 16 people, so the teams are quite small, but we have a catalyst team focused on unblocking constraints, early stage research in collaboration with Ag Emission Centre and others, and supporting some of the current programs, which I'll touch on later. Strategy engagement, work closely with the sector and investee companies to connect with farmers, connect with processors and regulators, and bring them to New Zealand, and a number of them are here, and you've heard from some already, and you'll hear from more about making sure they understand the New Zealand context of the work we're doing. And mostly what I'll cover today is our ventures team, who focus on the investments themselves and the early stage companies. And at the end, I'll talk a little bit about adoption and the challenges and opportunities associated with that. And noting here also, we are a strategic partner with the Ag Emission Centre, and we work very closely together. So mostly today I'm going to talk about this slide and the slide that comes after it. These are the tools that we're investing in, and you'll see it's a portfolio approach, a range of different types of technologies, knowing that there won't be a silver bullet, and that some of these companies won't be successful. If we could pick the ones that will be successful at the start, it would be a lot easier, but it isn't. So we've invested in a whole range of different types of technologies, and knowing farmers also want to have choice. Farmers don't like to be told what to do. They really appreciate the opportunity to choose what will work in their farming system. The last thing is we only invest in companies where the technology could work in a pastoral farming system. In some cases, that technology will need some adaptation to work here, but it must be able to work here. We take independent science advice before we make the investments, and we continue to take that advice through the term of our investment. So you see we've got a group of different types of inhibitors, Acteria Biotech, Ruminant Biotech, Agriceutical Products, and Ruminate, as well as the New Zealand Methane Inhibitor Program, and Zoetis is our partner with us in that, with Ag Emission Centre and Bioscience Institute. This is an area where we already know inhibitors work, but how do we apply them in a New Zealand context, different types of inhibitors. I'll also note we invest all over the world, so we've got investments in New Zealand and in Australia, but also in Europe and in the UK and the United States. And we'll invest anywhere where the technology is being developed that could work here. Also, when we invest, we require the companies to sign a side letter with us where they commit to bringing their technology to New Zealand and endeavouring to get it approved here by the regulator if that's required, and then making it available to New Zealand farmers. And we need to continue to invest to continue to hold those side letters. So as companies do further rounds of investment, we usually invest initially at seed and sometimes pre-seed for those who know the jargon. As we follow through with those investments, we need to keep investing to retain those side letters. So it's important we stay alongside them. But we also work with them to help them in the New Zealand environment. As I said, introduce them to the regulator. The regulator has been making great advancement, I'd have to say, in really understanding what's needed here, working very closely with the companies, making it clear to everybody what is needed to get regulatory approval, which I think has been very helpful, and really appreciate MPI for the efforts there. And then we have 2 vaccine investments, one in New Zealand, Lewis Frost Nbryo, one in Boston, RKO Bio, taking different approaches to vaccine development, both of them using natural antigen approaches, so antigens that already exist in the animal and applying those and choosing different antigens and working out the right mix and the right adjuvants. It's a long journey with vaccines, but progress is being made. So when we do get there, and I'm confident we will, they're a really good a tool for extensive pastoral farming systems where it's challenging to use some of the others. Biolumic, pasture-based product, adding lipid effectively to pasture. We heard yesterday that adding lipid to the diet of animals, improves the efficiency, reduces methane emissions effectively. More challenging, the pastoral system, Biolumic effectively does that by seed treatment, making really good progress with its New Zealand research. And then a range of probiotics. So these are the tools that work with the rumen to try and change the way that it works, improving efficiency, improving productivity, and also reducing methane emissions. We've got genetics, the sheep genetics we're co-investing with, Nbryo, who's here, and Armour, which is focused on nitrous oxide. Moving on, this is the timeline. So these, I'd have to preface this with, these are the data that are provided to us by the companies we have invested in. That said, we work very closely with them. We're often on the boards of these companies, so we do have a good understanding of where they're at. We're not being absolutely specific here because in some cases we don't have approval to release that information. But you can see here the rough estimate we're expecting, which clearly is a ruminant biotech bolus, to be in market this year, and then a range of others over the next few years. So by 2030, there should be a range of tools available, knowing some of these will not be successful in getting to market. And last but not least, since I'm getting the 1 minute board, the adoption. Now, we can't underestimate this. It's been talked about a lot. We need farmers to be able to use these tools for them to be effective. They've got to be available. They've got to be affordable. They have to be accessible, easy to use. There needs to be a range. There will need to be ways of reducing barriers. So early adopters using technology, farmers like to see what other farmers do, early and getting commercial scale out there. And also there'll need to be some kind of incentive. I mean, a number of these companies are working on productivity improvement as well as emissions reduction, but in the absence of productivity improvement, which is sometimes quite hard to prove, then there'll need to be some kind of incentive. And I know others have talked about that. And that is my time up. So thank you very much.
Thank you. Exciting to see that bolus getting closer, but also the full sweep of other things that are coming down the pipe as early as next year. I'm sure that none of the speakers to come today are among the companies that are not going to make it to market. I'm sure you're all going to be the winners. We now have a set of speakers who are going to give us a series of rapid fire presentations, each one on a specific product, 6 technology programs from across the portfolio. As a reminder, you can submit questions for all of these people via the Hoover app, and we'll get to these in the Q&A following the presentations. One final reminder about seats. If there's anyone looking for a seat, I can see from here there's about 15 in this area here. So if you're wanting to come and sit down, you're not quite sure where to go, this zone here has got heaps and heaps of available seats, just about a whole table right there at the front. Now, we're going to start with Lewis Frost from Nbryo. You see what they did there? He's going to kick us off.
Thank you very much. I'm going to speak a little bit about what we're doing and how Nbryo is reimagining the livestock breeding system for future food resilience. Now, I think we've heard a lot about some of the information contained on this slide today. Hayden from the Global Methane Hub did a great job of highlighting some of this information. So I'm not going to drill down onto where the emissions are coming from and what category of livestock. This is data from the Pathways report produced by FAO in 2022 and 2023. A lot of you will be familiar with it. I'll be quoting some other data from this source and can provide a link to it if anyone is interested. But regardless of this fact, that these emissions are coming from livestock and cattle in particular, livestock have such an important social, cultural and nutritional role in our lives. More than 1.3 billion people rely on livestock for their livelihoods, and they play such an important role in food security around the world. Being nutritionally dense means they're an important component of many diets, and we know that as the global population grows and many countries begin to improve economically, demand for animal-sourced foods, and in particular proteins, continues to increase. So as livestock are such an important enricher of our lives, we can't just seek to reduce livestock numbers to reduce methane emissions. We need to come up with novel and innovative ways to reduce these enteric methane emissions while seeking to improve productivity at the same time. One of the tools we have at our disposal is genetics and breeding. A substantial work has gone into this globally. Current breeding systems can achieve reductions, and selective breeding changes are cumulative and permanent. With current systems, estimates are that we can achieve between an 11 to 26% reduction in 10 years. So the good thing is we can make changes and they are permanent and cumulative. The bad news is it's just too slow. It's going to take us 10 years to achieve an 11 to 26% reduction. So we could be doing more and we could be doing it faster, we believe. This is some more of that FAO data, a waterfall graph looking at the bar circled in red being the predicted total 2050 livestock methane enteric emissions if nothing was done, and then sources of potential mitigation waterfalling or cascading out to the right. And I'll draw your attention to the two largest sources of mitigation, luckily for all of us in the sector, being productivity improvements and breeding and genetics. Two things that we can use technology to do something about, and that's what we at Nbryo have been working on. So I'll give you a little bit of background as to where we've come from, where we started, and where we're going with our technology platform. Nbryo started at a cattle breeding property known as Nindawimba in southeast Queensland, founded by Ewan Murdoch and Nick Cameron. For a long period of time, over 20 years, they were custom breeding bulls for large pastoral operations using a range of artificial breeding technologies. And they worked their way up to extensively using IVF and realized that IVF was the solution to the next breeding system in cattle. The challenge that they found was, how do we make it affordable and scalable and able to be implemented by the commercial producer right around the world? In 2021, they took that nugget, that core of IP they'd developed, they formed the company and raised their first grants from Meat and Livestock Australia, the Australian equivalent of Beef and Lamb, and the Gates Foundation. Subsequently, we've received support from grant funders and equity investors, including AgriZeroNZ. Our tagline is 7 years in 7 days. So where this comes from is we believe existing livestock breeding systems over time without enough change have become outdated and slow. And if you look at a really well-run genetic improvement program, it's probably achieving 1 to 1.5, some of the best in the world, 2% rates of genetic gain a year. It's going to take you 7 years to achieve that 10 or slightly greater than 10% change in a trait or a group of traits if we're looking at something like methane emission reduction. We believe that if you took all of the technologies we're developing, some are commercially available now, some are still in R&D, and you deployed them all in a 7-day IVF cycle, it would be possible to make the same level of change in a single 7-day IVF cycle. So 7 years in 7 days. This is the platform I'm talking about. Identify, scale, deliver, and analyze. Starting with identification, what we're doing here is non-invasively sampling the DNA and RNA of an embryo as it develops and running a whole host of genomic tests on that embryo pre-implantation. Any test that's commercially available today could be run on an embryo, so you're essentially drafting embryos in a dish. You're not waiting for an animal to be born. You're not waiting for it to be performance recorded. You're making selections way, way, way earlier than would otherwise be possible, including for some of the emerging markers around potential methane emissions. So now that you have your superior embryos, we use a proprietary technique we have around the multiplication of embryos in scaling, where we can take an embryo, we're not cutting it, we're not splitting it, we're not using blades. We stress these embryos and we get them to undergo the natural process of twinning that they would in utero 2, 3, 4 times. We've done this up to 21 times from a single embryo. So now you have many, many embryos from those superior embryos you selected. And we are offering this service commercially in our laboratory in Australia. And one thing it does is it gives you more of the superior embryos, and it also drives down the price of those embryos due to the scaling factor. So making them much more affordable and accessible, particularly for commercial producers. Now that you've got all these really good embryos, anyone who's run an IVF program will know one of the biggest challenges is getting a skilled technician with availability to put those embryos in. So what we've done is we've developed a digital device, the world's first digital embryo transfer device. That means anyone with rudimentary artificial insemination skills can navigate the cervix, detect what side the corpus luteum is on, and deep uterine horn transfer an embryo with no epidural and no deep rectal palpation of that animal. So it improves animal welfare outcomes for the cow and allows you to transfer your own embryos, further improving accessibility of IVF and embryo transfer on farm. From an analytical perspective, we're collecting data from cow to calf as we collect oocytes on farm all the way through the IVF cycle to the calf being ultimately produced at the end. The outcomes are lower costs, higher productivity, and reduced emissions. How are we reducing emissions with IVF? 3 ways. Through improving productivity with superior animals, we're generating a lower emission intensity. Through structural herd changes such as eliminating bobby calves, because every pregnancy has a purpose through highly selected embryos, and through genomic selection for lower methane producing animals as those markers start coming online and deployed in the identify step. So what's next for us? We completed our 2025 seed raise of 18 million AUD Australian, which AgriZeroNZ, we're very grateful, participated in. We have started commercialising the first of those embryo technologies in the multiplication technology, and we're looking for partners in laboratories, in field technology, and in genetics here in New Zealand to launch this technology. Despite looking like a pretty healthy cap raise, we don't have the capital to build laboratories in every market around the world. Our plan is to license this technology to in-market providers locally and allow them to service their own countries with their own expertise using this technology in market. If you'd like to know more, please get in contact with us. We'd love to have a chat. Thank you. And up next is Andrew Leech.
Thanks to everyone today for turning up, and thank you to the conference organisers for the invitation to speak. It's not forwarding. There we go. So what was Bovotica set up to do? So about 3 years ago when we were spun out, we had two really clear objectives. We wanted to develop a probiotic bacteria that used native rumen isolates, no genetic modification, that decreased methane. Now, we wanted to select these isolates so they didn't just decrease methane, but they also increased productivity. We've done our analysis in several cattle data sets. We believe we can do this. But as we've continued to develop our strategies around our technology, we think we can do one thing more as well, and that's improve quality. There's a growing body of evidence at the moment that shows the rumen microbiome is a direct contributor to improving quality traits in meat and milk. In dairy, this may be improving milk solid yields. So this is a research program we've got at the moment, and we think it could be a really good driver of value in the technology for the producer, along with the productivity gains we think we can deliver. So how do we design our probiotic technology? Well, we start with a big data approach. We've got these cattle feeding trial data sets. We segregate individual animals out into groups based on low methane, high productivity, and high methane, low productivity. We then do a comparison between the rumen microbiomes between these two sets, and that allows us to rationally select bacterial isolates from the rumen that can contribute to methane reduction and productivity. From there, we have to isolate these strains, get them into the lab. There's actually a lot of technical challenges that we've had to solve in that bit, but we've built a rumen biobank. I've got a slide on that later. From there, it's a fairly standard, what I would call a drug development process, where we do a preclinical assessment of our strains, we test them in bioreactors, we formulate them into products, and then we deliver them into animals. Initially in animal trials, and we've got a series of trials we'll need to run to get products on the market in various jurisdictions, but then eventually to deliver products to market. So this is one of the analysis that we have performed. This one is actually for beef cattle that are on a concentrate diet. So this is called a Linda plot. It's a differential analysis. On the x-axis, we're looking at methane and feed conversion ratio. So the units we're looking at here are grams of methane produced per kilogram of body weight added. On the y-axis, we've got bacterial species abundance. Each one of these dots is an individual bacterial species that is in the rumen. Now you can see from this differential analysis, we've got about 20 or so species there that separate themselves nicely from the rest of the rumen microbiome. On this graph, we want to select strains that are as far left as possible and as high as possible on the graph. This means they're having a bigger effect in the rumen at reducing methane and driving productivity. So let's have a quick look at the mechanism of action for how our technologies actually work. This is a simplified diagram of the digestion processes that happen in the rumen. I'll draw your attention to the pyruvate to butyrate and pyruvate to acetate fermentation steps. What you'll note here is there's an excess of hydrogen produced. So our strategy is to load the rumen with bacterial strains, our probiotics, that actually push energy, carbon, and hydrogen down those alternative fermentation pathways. So we're looking at pyruvate to succinate, pyruvate to lactate, and lactate to propionate. So if we can push additional energy down these pathways, you get a deficit of hydrogen being produced. The methanogens basically starve, their population shrinks, and methane goes down. That creates basically an addition of energy in the system, and then our probiotic strains are feeding that energy into the production of those other volatile fatty acids, and that's what drives the production efficiency in our technology. So where are we up to? We're currently very much still in the lab. We've got a lot of work to do. We've created our rumen biobank. There were challenges there, but we think we've solved all of those. We're currently assessing the probiotic candidates we have, and we'll be testing them in rumen bioreactors in the next couple of months. We're currently on track to do our first animal trial in August, September this year. That's going to be performed in Australia, just outside of Townsville, and that will be run by Ciro for us. That trial is a small group of animals. It will be conducted in respiration chambers so we can get those high quality methane measurements that will confirm our technology is at least working in the animals and reducing methane. From there, we've got 2 further animal trials planned for Q4 this year. We'll be doing a dairy productivity trial with Agricultural Victoria, and we're doing a beef productivity trial as well. And we're just finalising which partner we're going to go with there. That will provide that vital productivity data. We know from a lot of discussions with industry and particularly producers, initially uptake of our technology is going to be driven by productivity rather than the methane benefits. From there, we need to raise a Series A. We've got a series of steps to get product on the market in Australia. We need to go through the regulatory system. But today I'll focus on what we need to do in New Zealand because that's probably more relevant to most of this audience. So what we've learned from doing our analysis and selecting our probiotic candidates is rather than the breed of cattle being important, what's important is what that animal is eating, because that's the food source that feeds our probiotics. So to develop a technology that's going to be optimized for New Zealand, we need to start with analysis of New Zealand data sets. I'm happy to announce we're just on the first stages of embarking with a partnership around LIC. They've developed a fantastic data set and they've agreed to give Bovotica access to it. And I think, looking at the synergisms that could occur between cattle genetics and rumen modification through probiotics is really exciting. We'll do our analysis there. We'll select our New Zealand probiotic candidates. We've then got the same kind of pathway to work through to confirm that those probiotics are going to work in New Zealand conditions in the lab first. We've got several steps we need to work through. We're already working with ACVM and EPA. One of the things we need to get over is getting our probiotics into New Zealand because you've got quite stringent biosecurity laws, and that's so we've had some good conversations there and we don't think that's going to be an issue. Now, most of you will be aware with the legislative changes happening in New Zealand pretty soon, the Bovotica technology is going to be exempt from regulatory approval here, so we can get to market very quickly in New Zealand. That's great for us. It's great for the New Zealand industry as well. We'll still need to run some trials in New Zealand to show our technology is working in New Zealand conditions. That's scheduled, we think, for about the start of 2028. If that checks out, we'll start the commercialization process. And I think it's realistic. If everything goes well, we can deliver our technology widespread in New Zealand in 2029. Okay, I'm running out of time. So a quick look at progress in the lab. We've built that rumen biobank, 3,700 isolates. We're midway through sequencing it. We're getting really good diversity, a lot of unique species, but quite excitingly, we've already identified 139 novel species of bacteria that have never been isolated before. That's important because most of our probiotic strains we want to use have never been brought into the lab before. We've got about 100 different bacterial isolates we're assessing now. And through that process, we'll work out what our probiotic consortium is that we take on to further testing in animals. So I've spoken about the technology, but what's really important as well is the products that we're going to produce. So we're in a lot of different discussions with producers at the moment as we want to make sure the products we produce are light touch, fit into your existing animal handling practices, and don't create an additional labour or cost burden to apply them. In intensive and semi-intensive situations, we think a feed additive is really the way to go. Regular access to the animals, it can be put into their feed, they're dosed every day. We can see that being an enduring business model to deliver this technology. Animals in the paddock are much more of a challenge for a probiotic technology. What we're looking at here is a high-dose oral drench that you can you can give to the animal every maybe 2 to 3 months. We don't know about the period of efficacy at the moment, but that will have, ideally that will help reprogram the rumen and have those functionalities of lowering methane and increasing productivity while they're out in the paddock. And obviously looking towards the future with early life intervention. Early life intervention is probably the best time to program the rumen into a low methane, high efficiency state. So here we're thinking a drench might work fairly well. Thanks for my talk. And if there's any producers, come up and talk to me. I'd love to discuss our product selection and how it may, if it's going to work for you. Thank you.
Well, I'm going to talk to you about nitrous oxide, which of course is quite different to methane because largely nitrous oxide emissions occur outside the animal, unlike methane. Now, nitrous oxide emissions account for about 12% of New Zealand's total greenhouse gas emissions, and about 95% of that comes from agriculture, and 80% of it are largely coming from animal excreta. Nitrous oxide is a much more potent greenhouse gas than both carbon dioxide and methane, and therefore control of nitrous oxide is certainly important to us. So nitrous oxide makes up about 10% of the total global greenhouse gas emissions. In New Zealand, 80% of nitrous oxide comes from animal excreta, predominantly from urine. So reducing nitrogen moving into urine would be beneficial. However, we have relatively few options available for reducing this in our extensive grazing systems. And on top of that, New Zealand pastures are often high in nitrogen and higher in nitrogen than in fact ruminants require. And so this excess nitrogen ends up in urine. And if you look at that graph on the right, you can see the solid dots on the graph are from urine, and the open circles are from dung, which just shows that urine nitrogen levels are determined largely by the level of nitrogen uptake, and most of that ends up, unfortunately, in urine. So in our forage-based systems, what options do we have that we could consider employing to reduce this nitrous oxide emissions? I've got 4 listed here. Plants with secondary metabolites that divert the movement of excess nitrogen from urine into feces. Plants whose root exudates contain compounds that inhibit nitrification. Increasing the metabolizable energy, so through increased carbohydrate or lipid of pasture, which leads to more animal product per unit of feed consumed. And lastly, looking for pasture species with root systems that have complementary and or high rates of nitrogen uptake from soil. So let's just look at each of those in turn. There's a number of compounds that are found in some plants which could be very beneficial in terms of moving nitrogen from urine into dung or feces. Condensed tannins would be right at the top of my list. These are found predominantly in legumes such as Lotus caniculatus, Sulla and sainfoin, and are very effective in diverting nitrogen from urine. Hydrolyzable tannins are related obviously to condensed tannins. They're not abundant in a lot of our forages. One example would be sheep's burnet, but there have been very few studies looking at the benefits of hydrolyzable tannins. Oxidative polyphenols, now these are found in plants such as red clover. However, just in caution here, these need oxygen to be activated, and just remember that in the rumen, it's anaerobic. Saponins, now these are found in plants we know well, lucerne and white clover. However, results have been rather inconsistent in determining just how beneficial these are in moving nitrogen from urine. And lastly, glucosinolates. These are found in brassicas, and again, we have rather inconsistent results related to those. So let's look at then, the slide doesn't want to move on. Move on to these slides aren't moving forward. So, So sorry, that was the slide you were supposed to be looking at. This is the slide we are now looking at. Root exudates that contain compounds that inhibit nitrification. This has been well documented in tropical grasses, but it's much less obvious in temperate grasses. And just be clear that some root exudates, such as sugars, amino acids, and organic acids, can in fact increase nitrous oxide emissions by accelerating microbial denitrification processes. Other root exudates, however, like phenolics, terpenoids, and flavonoids, can significantly reduce nitrous oxide emissions. There are a couple of species of interest here. Plantain, it can reduce nitrous oxide emissions by up to about 40%. And fall-based pastures, which obviously include plantain, but also maybe chicory and some lotus and grass and clover, can reduce nitrous oxide levels by about 19%. Having all sorts of trouble moving this forward, right? Looking at increasing metabolizable energy in pasture. Now, grasses containing increased levels of carbohydrate can reduce nitrogen excretion to urine by about 29 to 30%. Increasing dietary carbohydrate intake can also be shown to increase nitrogen excretion into feces by about 76%. So that is very beneficial. Similarly, increasing lipid levels in plants can have the same effect as carbohydrate in terms of moving nitrogen into feces. There has been one project looking at high metabolizable energy perennial ryegrass, which could lower nitrous oxide emissions by about 38%. But just note that that is a genetically modified plant. So if we move to root systems that have complementary and or high rates of nitrogen uptake from soil. So reducing soil N levels through increased plant absorption could lead to reduced nitrous oxide emissions. As an example, Poa trivialis or rough bluegrass and perennial ryegrass together reduced nitrous oxide emissions by 44% in one study compared to just perennial ryegrass alone. And similarly, if you have grasses such as Italian ryegrasses that absorb nitrogen at lower winter temperatures, they also reduce nitrogen leaching when the soils are very wet, and concomitantly, indirectly, nitrous oxide emissions from rivers and estuaries. So look, in summary, forage-based solutions to reduce nitrous oxide emissions are needed for situations where ruminants are grazing outdoors year-round as they do in New Zealand. There's no easy and ready-to-use solution, while some opportunities have been identified. And for me, the top three options and their challenges include plants with condensed tannins. Now, the issue with a lot of those is agronomic suitability in mixed species graze swards, or the regulatory hurdle where we actually, in fact, have got condensed tannin expression in leaves of white clover, but again, it's genetically modified. We have been trialling these in Australia under license, and they look a very promising option for us, but again, we need to go through a regulatory process. Forb-based pastures, so this includes your plantain, chicory, maybe some lotus. These are gaining farmer acceptance to integrate in their pastures, but they will need specific management systems for them to be actually effective and to persist. And lastly, biological nitrification inhibitors. We need much more research in this area to determine just how these would operate and how effective they would be. So in conclusion, the motivation for pursuing any of these will be driven by recognition of the responsibility to reduce greenhouse gas emissions, but they need also a concomitant increase in productivity and or regulations, which I think won't go down well, that demand compliance. So thank you very much.
Good morning. My name is Jake Bubolz. I'm from Zoetis, Senior Principal Scientist. And I just here to talk about the methane inhibitor collaboration that we currently have with AgriZero and Ag Emissions Center. So really, I want to just also call out our collaborators here with BSI, who's doing a lot of the ag research, directly, and from the early perspective, and also calling out the Ag Emissions Center and AgriZero in Bodie. Before I get started, I just want to kind of go back to a question that was raised yesterday, and that was a New Zealand argument is that we're too small to make a difference. And carrying on to that is what's the international community perspective and view on the emissions, on the methane emissions direction. So really, I'm here today to really lay that out and say, although I can't speak for my government, I can speak specifically for Zoetis, and saying that we are definitely invested in sustainability, and that's the direction that we're moving forward. And that's a direction that we're showing today. That really, there's three aspects to that. It's watch, listen, and go to the experts. So with that, we're collaborating directly with the AgriZero and BSI, and we're advancing a product. With that being said, we're really looking right now at, Zoetis is a leader. Well, Zoetis is definitely invested in sustainability, and that's no surprise. We're with our animal health and productivity products. We're definitely striving to give the best productivity of the animal by really focusing on the health of that animal. So in partnership, we're really looking at a small molecule that we're driving forward for a methane inhibition product directly with BSI, and it's a targeted solution. So we're not looking at disrupting what's going on in the rumen, we're enhancing what's going on in the rumen. With that being said, we are looking at and directly affecting the methanogenesis approach and reducing the overall methane, and without compromising animal performance, and really looking at enhancing that performance overall. With that being said, really we're looking at a flexible, sustainable solution. So we're looking at something that has delivery pathways in feed additive with premix or a slow release device to really target those solutions across across the NZ landscape. There we go. So right now we are in a stage where we're working directly with BSI, and the first approach is really identifying what's out there. BSI has been working at this for a long time. We're at the first stage was in silico screening. So we started with over 15,000 or 15 million molecules screening against those compounds against in silico screening. And from that, we got down to 140 against specific 8 enzymes, and narrowing down to 40 molecules where we did methanogenic culture. And then from the 40 down to 13,000 molecules, which seems like a smaller number overall, but still a lot. But over that was tested in the rumen in vitro assays, which is a fluid-based assay. From that, we took the best that are most efficacious across the board in vitro systems. And we narrowed it down to the top 2 classes. With that being said, we're pairing directly with the BSI, and we're providing support. So additional screening, initial screening was all done by AgriZero and BSI. Moving forward, we're really pairing as we can bring the commercial expertise and the safety expertise too. The big component here is really going to be the de-risking. So up front, we're doing a lot of testing in silico to in vitro to in vivo. That's why we really want to get across with this slide, that we're de-risking these components, that it will take more time up front, but in the long term, we're looking at success and expediting once we get to the time of development. So in these early initial screens, we're also looking at toxicology and environmental effects, what's going into the environment, what's going into the animal, what's hanging around in the animal, what's getting metabolized. With that being said, we're moving forward with in vivo testing with the 2 top classes and looking at safety and efficacy demonstration in both sheep and cattle trials up to 56 days up to this point. So how we've been screening it so far is that we've been leveraging the expertise at BSI, running it through sheep chamber trials and then into cattle trials, either in chambers or in the US, where we have systems also in place where we can do in-feed mixing. We do hand mixing up front and then in-feed mixing in commercial-like systems. So we do early testing to get that understanding right off the bat. We have seen efficacious molecules. We have the top 3 candidates within the top 2 classes. And once we have the top candidate selected, we'll be looking at a longer trial at the end of this year. So we're progressing, but the big thing here is that we are de-risking this up front, so we understand exactly the implications that could arise, at least if there are flags, and how we can mitigate those risks in the long term. Once we have the final candidate selected, we'll be moving forward with a slow release device and really looking at the evaluation and progress in parallel with the top candidate assessment. So we'll push forward with an in-feed aspect of it, but also a slow release device once we have the final candidate selected. Once we have that selected, we'll also be looking with a project development plan and regulatory pathways. So we are in connections with the different regulatory authorities across the landscape, since we are a global company and we're looking for global solutions. That's one of the aspects that we will be leveraging. And we have early discussions, but once we have that final candidate selected, we'll be able to better map out the trajectory. On-farm application. So the big thing here too is flexibility. So we want to allow for both the TMR, ration controlled aspect of this, with the straightforward daily inclusion. Also the pasture-based systems, looking at the slow release device, but we're also optimizing the formulations. We'll optimize those formulations once we have a better understanding of exactly the direction we're going with the final candidate. The fit with operations is also extremely important. We want to make sure it works with the current management practices. This is for the producers, so we want to make sure that it works with genetics, high and low. We actually have genetics at Zoetis that we're looking at within the cattle in the US and looking at the effects within those different groups, also with other feed additives and management changes. So this is an additional aspect to it. We talked about stacking throughout the conference. That's something that we're also looking at. Really the bottom line is the ROI. So we're really looking at the balancing of the dose for optimal methane reduction, but also animal productivity. So we want to make sure that we really are looking at the bottom line for the producers and the best benefit for them as well. All right, so the planned actions. I know everyone wants a product yesterday, and we're working the best we can to get that out. But like I said, we are really looking at de-risking the product overall. So once we get to the stage where we're having conversations with regulatory authorities, we can blaze a path. So really right now, we're confirming the dose selection. And once we have a final candidate, we are confirming the doses and also the withhold verifications. With that, we'll also accelerate the NZ relevant slow release devices. And then we'll be looking and implementing larger trials across regions, seasons, and breeds. The one thing we can hang our head on right now is that we do have trials that are relevant to New Zealand going on in New Zealand within the chambers and New Zealand cattle, also with US cattle as well. The big enablers here are going to be the clear regulatory path, delivery options to fit on-farm practices, and really looking at those processor programs and how we fit as well, and with the MRV devices. So we'll continue to assess those and make sure that we have something applicable and usable for farmers. And just want to also thank the BSI and the AgriZero contacts and Ag Emission Center. And these are, if anyone has any questions, please ask Wade. He is here. But thank you very much. I appreciate your time.
Good morning. I'm Richard Spelman from LIC. I'm just here to give an overview of our dairy cattle genetics breeding program that we've been researching for methane. So some of the work that we've been doing here is really over the last 5 years. So with methane for us in the dairy cattle system, and actually across all production systems, there's really two sides to it. One is really around emissions intensity, so the amount of methane that's produced per kilogram of product, and the other one is the gross emissions. So genetic gain, what we do pretty much every day as part of the breeding program, adds about 1% of productivity per year. So Cameron Baggery talked about yesterday, about 1.6%. In the agri sector, certainly in the dairy sector, about 1% of that comes from genetics. So the impact of that is that we're actually reducing the amount of methane per kilogram of product by about 0.7% per annum. So that's happening. But we want an and. We also want to reduce also the gross emissions. And Melissa talked about this morning, the amount of feed going through a rumen. We know there is genetic variation for the amount of methane that's produced for every kilogram of dry matter that is eaten. And that's what we're looking at with this work. So we've been doing this, as I said, over the last 5 years, working very closely through the funding from Ag Emissions Centre, also working with CRV, another dairy cattle breeding organisation based here in New Zealand and also the Netherlands. So we've taken an approach which is a little bit different. The approach has been really around from an adoption point of view and also from a cost perspective. So within the dairy cattle industry, between LIC and CRV, we generate about 85 to 90% of all of the replacements that are taken through the dairy system. The other side of it is it's very expensive to measure methane. A lot of you will know already. For us in the dairy cattle side, it's about $3,000 to $5,000 per animal. So we had an opportunity that we saw with our ability to use genetics from both LIC and CRV, that if we measured methane on our young animals, on our bulls, in this case, this is not a sex-limited trait. The other things that we work with, milk production, fertility, we can only measure them on females. So here was an opportunity to use a very small pool of animals between LIC and CRV. We purchase about 300 bulls per annum. Could we measure methane in those animals? And would that be predictive of where the most of our methane emissions occur, which is in lactating cows? So we've gone through 3 stages in this experiment. One, firstly, measuring young bulls. Could we measure methane? Is there genetic variation for methane? And then if there was, we would generate some heifers, measure them while they're growing. And then the third piece of work is where we are at the moment, which we've just finished, is measuring those same animals as lactating cows. So the early results have been very encouraging. We have measured over the last sort of 5 years over 1,000 animals. And after a couple of years of research, we decided that we would select the high methane size and also the low methane size. You can see here on this graph, there's a reasonable separation between the two groups. So we used 25 bulls in the high group and 25 sires in the low group. We worked with PAMU. We inseminated a herd with PAMU, and we generated about 400 female or heifers from that group. They came into our facility when they were about 12 months of age, 10 to 12 months of age, and we measured those two groups as well. And this was presented the last time we met here at the conference. We were very encouraged to see that as growing heifers, we also saw quite a good differentiation between the heifers that were high and low. Now, we only would expect that half of the difference that at best that we see in the sires would be passed through to the daughters, because only half of the genetics of the daughters comes from the sires. So very encouraging outcome from this work. The next challenge is really around what would happen when these animals are lactating. So we've measured our animals for methane and feed intake. Feed intake is very important for us. We don't want to be breeding an animal that just produces lower methane because she eats less. So we measure that over 5 to 6 weeks. We also measure milk production. At the same time, we've also been taking other measurements off these animals. So we've been using these portable accumulation chambers that AgriZeroNZ and BSI have, the rumen samples that will go to Bovotica, and also at the same time, looking at milk samples. Is there different fatty acid profiles that we can use or mid-infrared spectra which identify high and low methane animals? And the reason for this is in future, are there proxies to measure methane rather than these quite expensive experiments? So the good news really is that when we do the lactating cows, we see genetic variation which was greater than what we saw in the young growing animals. So we're seeing heritabilities about 20%, which is in line with international studies. However, probably the result which we've come across in the last month, which is slightly disappointing for us, is that the difference that we saw at a growing heifer stage is not replicated as a lactating cow. So as you can see there, on the left-hand side, that's what we observed. That's there is showing over the 4 weeks in the experiment. We saw quite a difference in amount of grams of methane per kilogram of dry matter eaten. When we go to the lactating animals, we don't see the same differentiation. There's no statistical significant difference between the two groups. So that's that result. We also, as an organisation, and also within New Zealand, a lot of our beef animals are coming from the dairy sector, so beef on dairy. We still have confidence that what we're seeing in our young bulls that we generate methane data for, that will follow through and flow through into these animals, these dairy or the beef on dairy animals in the production system. However, back to dairy, the next steps for us. So what we're seeing is that the trait that we see in a lactating cow is genetically different to what we're seeing in a growing animal. And so what we need to be doing is in future is measuring more and more animals in that lactation phase. To date, we've measured 300 animals. So it's a small group of animals. We'll have more data coming through in time, and that will continue to test that hypothesis that we had around measuring young bulls or that predict lactating cows. But at the moment, we don't see that that will be a pathway for us. However, as I showed, the genetic variation that we're seeing in lactating cows is certainly giving us an opportunity for us to see that genetics can play a part. However, to get to that outcome, we need to do a lot more measurements on lactating cows. We also need to find different ways through proxies or other measurements in a cheaper way that we can measure methane, because we can't measure thousands and thousands of animals at quite an expensive cost with the green feeds and feed intake. So we will need to do larger scale trials in the future, but we do see genetics as being one of the possible solutions. So genetics has always been a long game. These results show for us at the moment, it's probably going to be a touch longer before we get there. Thank you.
Hi, everybody. It's really great to be here. Well done for hanging in there. I am the last speaker in this session, and I am going to be talking about methane inhibition and electron flow. So we're going to be talking about fundamental science, and I know with a title like that, you're all super excited for what I'm going to talk about next. Okay, so let's get into Chief. Right, some context. So methane inhibitors are commercially available and they're in use globally. They haven't quite reached on farm here in New Zealand, but from what you've heard over the last 2 days, they're on the way. When a lot of the work that's been done in the sort of scientific literature in relation to methane inhibitors is being done around efficacy trials, and there's really good evidence behind some of those leading compounds. But of course we know, and those efficacy trials tend to last somewhere between 3 and 6 months. But of course we know that our animals here live a little bit longer than that. And so if you looked in the literature right now and you said, do we have an animal trial from a peer-reviewed publication that's looking at over 12 months of application of these types of inhibitors? There's one, right? So that seems a bit crazy. So there's a gap there that we need to look at. There's also two very big fundamental questions that we still need how we can use methane inhibitors to try and resolve. One, it goes back to, and I think I can say last century, when scientists actually made this hypothesis when they did some of the thermodynamics work, is that methane is a loss of energy for the animal, somewhere between 2 to 12%, depending on how you do your calculations. We now have methane mitigation techniques that can create a rumen-inhibited animal, but we're not actually seeing that increase in productivity, or it's not always predictable. It's quite inconsistent. And so that's telling us that we really don't know at the level we probably should need to know about where is that energy going, because of course we really want to try and develop technologies that are a win-win for our farmers, good for the environment, but also good for the farmer. We want to be able to make money or save money. A second critical gap that we have in this space is, we all know the story of antibiotic resistance, use of antibiotics, it's in human and also in animals, we've seen that rise of antibiotic resistance. As a microbiologist, microbes are pretty smart, they're pretty good at evolving, evading, changing it up. And so we really need to look at longer term studies to understand what the impact, how is the host responding, how is the microbes within that host responding to long-term application of these types of compounds. So CHIEF is essentially, at a high level, it's just an animal trial, which is being designed with the relevant measures to address some of those questions. What's unique about CHIEF is normally when people do, as I said, those efficacy trials, they'll often bolt on a microbial component. But of course, if you're asking a microbial question, and of course, methane formation is microbial, you need to design your animal trial slightly different. And this is what CHIEF has done. It's focused on the methane formation process first, then designed the trial and created the power in that trial to be able to answer the questions we need to answer. It's not Nobel Prize winning science. It's still really important. What are the main barriers? Why have we only got one publication out there? Well, there's two critical barriers that we've struggled in the scientific area to get over. One, it's the cost of these trials. Just like the cost of living, the cost of doing this type of trial is expensive. Also, it's challenging. There's no one area that probably has all the specialist capability that you need to do this type of research. And I guess this is where, the funder coordination, particularly at a global scale, starts to come in. We heard Hayden talk this morning about the Global Methane Hub, Enteric Methane R&D Accelerator, and we've also heard, of course, Naomi talk about the Ag Emission Centre, both two public good initiatives. They've been able to come together and partner to fund this trial, which is sort of removing that barrier of cost. And I'll also talk about how they've brought together the capability to do this type of work as well in further slides. Alongside this, both of these are public good. So all of the information that gets generated under this type of trial gets put out into the public domain to be used by whoever needs to use it, particularly in developers who are looking to develop these types of technologies. And three, I think it's important to note, funder coordination has happened. This is just one of 3 trials that is going on globally. There's also, while the New Zealand trial is firmly based in sort of fundamental science, it's not reflective of a practical type of application. There are 2 other trials going on, one in the States looking at 3-NOP, another in Australia looking at bromoform. And they're looking much more at that productivity, animal welfare, in a commercial, setting. So what does Chief look like? It's basically got three groups. We've got our control, and we're working with two of the leading sort of methane inhibitor compounds that are currently out there. We've got 3-NOP, and we've also got bromoforms. There's 16 animals per group. And important for this type of work, we need fistulated animals. And I think Matthias yesterday showed some pictures of fistulated animals. So these are animals that allow the scientists to get access to the samples that they need to be able to do that very detailed thermodynamic type work. The trial has already started. The ladies entered their accommodation back in February. We've already gone through the first measurement in respiration chambers. And they've also had their first sampling session, and they'll carry on for the next year. You know, they'll be sampled 8 times, a pre and a post, and about 9 months of application of the treatments, which are starting momentarily as we speak. The project team, like I said, often this can be a barrier to this type of work. So it does take a global effort here, and this is where I think funder coordination plays a very important role. And we've already talked about the Global Research Alliance and their network of scientists. So, you know, I'm not going to go into what these institutes are doing over the next year. You'll hear from the scientists about this particular work, but the base trial has been done here in Massey and BSI up in the beautiful Manawatu in Palmerston North. Shout out to Pammy. But also the analysis, a lot of the work is actually being done by specialist labs around the world, done here both in our Rumen Micro Lab in New Zealand, but equally we've got 2 specialist labs coming from Australia, 2 in the States and 1 in Ireland. And these really are the who's who in terms of, you know, it's a very robust, incredible team to sort of bring together to work in a consortium. So I'm really looking forward to seeing to where this project team gets to. So where do we want to be? You know, if we come back to this conference in, you know, hopefully in 12 months, what would we like to be seeing coming from this type of work? I think, like I said, you know, we really need to get data that can help us understand how we can get that win-win situation. Like I said, regardless of what you see in the media, when you methane inhibit it, you know, there's very limited data around increased productivity. But according to thermodynamics, there is the potential there. But we need to understand a lot more at a deeper metabolic and biochemical level about where is that energy going, because only when we know that can we then start to do learn to sort of modify it or manipulate it into that win-win situation. We're also going to, of course, have a very nice data set. You know, 9-month application of these inhibitors. You know, we're going to have to start looking, and we'll be able to use that to look for any red, you know, flags. You know, with microbes, I know from all my years, you've got to be one step ahead of these guys, right? You've got to be figuring out how are they responding to this type of, you know, how is the animal responding to, you know, being a low emitting ruminant through an inhibitor. And of course, the more knowledge you have on that, then the better you can in terms of managing it on farm. If you think about how we manage parasite resistance with sheep, you know, it's obviously a rotation of these types of inhibitor type compounds. And with that, I will stop there. Thanks, everyone.
Thanks, Sinead. Could I get our other panellists to come and join us too, please? We've got lots of time for questions, which is good because we have a lot of questions to get through. Now, look, some of the Palmerston North comments are starting to feel a little pointed. I hope they're not directed at me and the shade I threw on Palmy on the first day. And look, there are no Nobel Prizes for this kind of work, but there should be, right? Like this long-term gap-filling, chipping away research is what we need, because we need to know why are lactating cows not responding the way we thought? Where is the missing energy going? This stuff's all going to matter if these technologies are going to bed in. Yeah, without further ado, I have a clear top question, and I think people have been seeing the media reports from the UK of people tipping their milk down the drain in response to feed additives being used. This question is more New Zealand Pacific though. How much work has been done to understand how consumers will respond to interventions like vaccines?
As a scientist, I'll answer this first. In terms of the science domain, there's been quite a lot of work done around, particularly in the States, in relation to consumer acceptance of methane inhibitor type acceptance. And there's actually peer-reviewed publications around that. You can access that. In terms of methane vaccine, I haven't seen anything in the peer-reviewed publication around how consumers react, but certainly from surveys, rural decision making, I think between a methane a vaccine and an inhibitor, I think what we know is that some farmers it's perfectly okay. For others, it isn't. In terms of consumers, I don't think the work has been done here in terms of the scientific literature, but it's perhaps something that industry can talk about in terms of their consumer analysis. And yeah, Jacob's nodding, so
Yeah, I can add a little to that. So from Zoetis's perspective, the way we're handling it is really market preparation as well as information. So I think generating that information up front and being able to supply that to producers and to the general public is really important to get their buy-in. I think a lot of it's misinformation, not understanding completely the full breadth and depth of the information provided. So that's how we're handling it. There's absolutely not really a lot of information out there when it comes to peer-reviewed journals, specifically on the inhibitors that are currently out there or the vaccines. It's evolving, but that's definitely something we're doing with our market research as well, and understanding how we can influence it and how we can best prepare the markets.
Just to chip in with something John Roche told me once in response to the Bovaer, the third option, experience in the UK was that there was a lot of heat, a lot of media, but sales did not measurably change. So I guess if the market is the ultimate kind of focus group, perhaps that one didn't pan out quite as badly as it could have. Now, the next question is, stackability has been talked about a lot, but what research has been done to determine the compatibility of different mitigations with each other, i.e., will inhibitors and vaccines alter the effects of feeding options, e.g. tannins and vice versa? I mean, I probably would call that research into stackability. Has anyone got a perspective on that one?
Oh, it's coming to me. I think for a long time, we didn't have anything to stack. And so I think that's the gap in the scientists. I think now we are starting to see, how, and there has been some work done with, for instance, the high lows and stacking it, if you want to call it stacking it with, a low methane named forages. And in that instance, they have seen some additivity, although very, very small numbers. Obviously, we don't have a methane vaccine to stack with something right now because it's still in a proof of concept stage. In terms of methane inhibitors and genetics, I don't think anybody has stacked those. Yes, you can stack them in terms of we've measured this is a high animal and this is a low animal, and people have done some trials with inhibitors with that, but it's not necessarily that they're true high and low, because you've obviously they may change when you change the diet. So I think the reason we haven't seen the stacking yet is that we haven't really had the technologies to stack, and that's certainly, I think where the research is going now. But I think it's important that not all mitigation options that we've talked about can stack, particularly an inhibitor and a vaccine. They're both targeting, directly methanogenesis, so and methane, those methane forming microbes. So they're both going after the same target. So it's likely, in that instance, it's likely to be one or the other rather than them going together. I think still a big question around how high and low genetic animals will respond to inhibitor, and whether it'll be cumulative. I think where we want to go with stacking is a little bit more about where the Bovotica story is going, is around, it's not about stacking to get increased mitigation option. often it's more stacking to get that win-win situation, and getting methane mitigation plus a co-benefit. I think that's where we need to be going, in terms of when we talk about stacking, or in my opinion anyway.
Yeah, thanks, Sinead. You kind of stole my input there. At Bovotica, we're really excited about how, with the probiotic technology, we've got the ability to stack with a range of different technologies. We're having conversations with all the other companies in the space. We're a bit too early to start testing, but once we've got some of our MVPs formed, we'll definitely be looking at how our technology interacts. You could see where you dose an animal with an inhibitor, that gives an advantage to our probiotics once they get delivered to the rumen. I like what we could do with vaccines, and obviously, genetics as well. So we'll be testing all those solutions, but it's to come in the years down the track.
I can well imagine. We're talking a lot about methane, but surely we can stack nitrous oxide technologies, carbon soil technologies, peat management, because they are operating on different things.
Correct, they are operating outside the animal, but in fact, a lot of the mitigation is going to occur by the way in which nitrogen is directed within the animal. And so I'd like to think we could develop forages that our animals eat, which enable that direction of nitrogen into the feces and away from urine. And I'll just also make the point that some of those compounds that I mentioned also will reduce methane as well, particularly the tannins, the condensed tannins and hydrolyzable tannins. They will have a direct impact on microbes in the rumen, and direct that energy flow away from acetate and into propionate, reducing methane emissions as well. So it's a bit of a win-win, but the trouble with these condensed tannin containing plants is they're not easy to grow. And they don't necessarily suit our competitive ryegrass, heavily grazed systems in New Zealand. We would have to develop other methods, just be aware of trying to graze our pastures to allow these plants to actually establish and express the potential that they have. The flip side of that is we could potentially do it in our dominant species such as white clover and perhaps even perennial ryegrass, but in some cases they may end up being genetically modified, so that it's an additional complication.
That leads us beautifully into the next question, which is, how do we think dairy companies are balancing the use of innovative technologies like inhibitors, vaccines, and IVF, GMO would be another one, with growing consumer demand for natural production and clear supply chain disclosure? I mean, perhaps a question embedded in that is, do we know what consumers consider to be natural and not natural? I would love to hear the panel's thoughts on this.
I think a big challenge of it is education. So an example would be IVF. There's probably a whole bunch of people in this room, paraphrasing, but who've had their own children born through the IVF process. Before they did it, it was probably scary and really unnatural. Once they got out the other side, it was probably a bit of a journey, but they're probably really happy that they had a child out the other side of it, and it was probably something that really helped them raise a family. So if you've never used IVF on farm, it can be a little bit scary and intimidating. Once you've gone through the process and someone's held your hand, it becomes something you can routinely use to improve productivity. So I think the same thing goes for consumers. It's fear of the unknown can sometimes drive some of that concern.
Yeah, any thoughts on that one? How we think that's being balanced and whether we can read the tea leaves?
I think you'd have to ask the dairy companies.
They are admittedly not on stage right now. Ooh, here's a goodie. For farmers needing to report their emissions, either voluntarily or to support climate-related disclosures, do we have sufficient data, methodologies and calculation tools readily available to capture and recognise the emissions reductions of farmers that are adopting these practices and technologies, or is the lack of these a barrier to implementation? So I guess, do we have provability in a way that will be accepted by the market and accepted by regulators is probably that question.
For genetics, I think it will be straightforward. I mean, so most of our animals, the vast majority, over 95% of animals are recorded through software. We calculate breeding values for all of the traits of interest. So if we had a trait for methane, I think it'd be very easy through the genetic evaluation. And again, we have a lot of animals that are DNA sampled at birth, about 35 to 40% of animals. So through genetics, I think we can certainly identify and predict their breeding values. We can verify it. So I think that has, in dairy cattle, a very easy system and very little extra work from a farmer at all.
Yeah, I guess I should bring in, I suppose, if we go back to, and I don't want to see anybody throw anything at me, but back in the Hewaka Kanoa days, you know, people did do quite a lot of work looking at the on-farm tools for looking at how you do MRV sort of on-farm. And since then, you know, there's, you know, what they found is actually some of those tools are quite useful. But, you know, MPI created the on-farm tool, which I think is hosted on the Ag Matters website, which is from a farmer perspective, you know, that's looking at, you know, the calculations behind that are very much in line with what we do in terms of our national inventory. So MRV on farm, I think we've got some good tools there. And over time, they are starting to add in, you know, mitigation technologies. Things like EcoPond obviously has gone right through all the way to the national inventory. So has nitrification inhibitors. So the code behind how you do that on farm is actually there and is in line with our national inventory. And as more technologies come online, I'm sure the code behind them will figure out. Some are much easier to do, you know, so, you know, in terms of MRV, something like a methane vaccine, you know, you can just look at the receipt and know that, you know, a farmer has bought it. Okay, there might be questions around, did you implement it? But, you know, it's quite easy to record. Something like the amount of plantain in a pasture and trying to verify that an animal has ate a certain amount, that's quite difficult. But there are people looking at technologies to sort of account for that as well. So, but that is much more difficult. So some are more easier, some are harder, but you know, people are thinking about how that, because at the end of the day, we talk a lot about emissions intensity, but you know, here we're talking about, you know, it's absolute emissions, right? That's the issue with the climate. So yes, you know, emissions intensity is a very good conversation starter for climate change. But at the end of the day, we need to verify, particularly for New Zealand, that we're reducing absolute emissions, because that's what our targets. And I think the Australian said we don't want to talk about targets, but you know, we still do have targets and international commitments.
It's going to be fun to watch those algorithms deal with stackability, isn't it, in the future? So glad that is not my job. Specific question about embryo. When you scale the embryos, are they genetically the same animal afterwards? And if you find an issue later in life with that cow, will you find it in all of the cows with that same genetic material?
So this is one of the benefits of testing the source material before it goes through to multiplication. You can test for all of the genetic conditions and genetic diseases that are available currently. So you want to interrogate that source material beforehand. And currently we're turning an individual embryo into, say, 3 or 4 embryos, and they come out as twins. So they are identical twins from that source material, and they will present that way. And we've grown them through to live calves and studied those animals on the ground for several years now. So they do come out as identical twins on the ground.
Question for John here. We did actually touch on this, but let's get a super clear answer. Which of the forage options for nitrous oxide reduction have co-benefits for methane reduction?
Well, I mentioned the tannins, condensed tannins, and possibly also hydrolyzable tannins. They haven't been studied as much as condensed tannins, but there is good evidence that both those types of tannins will reduce methane, as well as direct nitrogen away from urine in the animal. But I come back to the point that these plants, while they are available to us, aren't widely used, and they do require different types of management for them to be able to persist and grow in our pastures. As for the other compounds I mentioned, saponins, there has been a little bit of evidence that they do reduce methane, but the results can be rather patchy as well, and it's just not definitive enough to be absolutely sure that that is a way forward. But I'd love to see our pastures, ryegrass and white clover, being able to express particularly condensed tannins, possibly hydrolyzable tannins, but definitely condensed tannins. That would make a huge impact on both methane and nitrous oxide emissions in New Zealand.
A Bovotica related question. The knowledge generated from low emissions genetics suggests a role for the host controlling which rumen microbes thrive and what they do. Are you considering host-related effects to enhance the establishment of your selected probiotics? I'm thinking about myself going to the fridge and taking a probiotic now and my host role in that whole process.
Yeah, the quick answer is absolutely, yeah. Right from day one, the technical team was talking about cattle genetics, how they may affect our probiotics. We want to know how specific candidate probiotic strains will work in, genetically optimized cattle versus non-genetically optimized cattle. So yeah, there's absolutely, I think for our technology to be successful, we have to look at all the different inputs that are contributing to what's controlling the rumen. So while we can control the species we put in, we can de-risk it by looking at what other factors are controlling whether they're going to persist there long term, other competing factors as well. So yeah, very important.
Another question for John. Grape pomace, I hope I'm saying that right, from winemaking is a serious waste issue. Given the condensed tannin opportunity you've talked about, is there a solution here to look at cross-sector?
Yeah, absolutely.
Grape
pomace is full of condensed tannins, and in fact it is manufactured overseas into animal feed, particularly added into concentrates. So for barn-fed animals, absolutely fantastic solution. For our grazed animals and pastures, not so good. And the issue is that you just can't give a dose of condensed tannins to the animal and it'll do for the day. The tannins have got to be there as the plant material is eaten. So the tannin is actually binding the protein and stop it being broken up and releasing the hydrogen, which then combines with carbon dioxide to form methane. The two have got to go together. So you can't just dose an animal with condensed tannins and say that's done for the day. It's got to be there in every mouthful. So for barn-fed animals, perfect solution. Grape pomace is a great way to go. But for our grazed animals, not so good.
Yeah, a concentrated bolus isn't the way to go if you're trying to deal with large amounts of waste, is it? And it's kind of... Another question for Lewis. What is the success rate of embryo implantation to calf? Are there any effects of the multiplication technology on postnatal attributes? I'm not 100% sure I understand that question. I'm hoping you do.
Yep. So it's probably primarily around pregnancy rates. We don't always get feedback on calving rates. We generally get feedback on pregnancy rates, and pregnancy rates in IVF range anywhere from the lowest would sort of be averaging 45% up to people who have been doing a lot of it and have a lot of really good experience and are dialed in, getting sort of, 70% is sort of the pregnancy rates they're getting from IVF. We're not always getting the exact calving rates. I think the second part of the question was, does multiplication have an impact on it? The data we're seeing in terms of multiplication's impact on pregnancy rate is a depression of 1 to 2 or 3% in terms of comparable pregnancy rate of intact embryos. So if you look at the extra number of embryos you're getting and the reduction in price point of around 20% reduction in price, it results in much more and much cheaper calves on the ground is the trade-off in technologies.
An LAC-related question here. This one I do understand because I was wondering it myself. Is what you're saying that the low emissions traits are present in the growing animal, but they're not linked to lactation and milk production? What's going on there where it's not translating, I think, is the gist.
Yeah. So what we're saying is, so what we've seen in our data set, we have to be very clear, it's in our data set, and others may have seen different things in their data sets internationally, but the genes that are controlling methane production in a growing animal do not appear to be the same genes that control methane in a lactating animal. So there still are genes that are controlling methane production from a host perspective. I mean, we're seeing 20% of the variation that we see of methane production being controlled by genetics, but it doesn't have a genetic correlation. Well, it has a genetic correlation of close to zero between the genes that are in a growing and a lactating animal. So there has been quite a bit of work, not quite a bit of work, there will be quite a bit of work and discussion really around why. No, end of the day. So the animals are in quite different physiological states. I mean, a lactating animal eats a lot of grass, well, in our situation, so it has a really significant feed intake. The animals go through at different times, different energy balance as well. And so there is quite a different physiological state that the animal is, especially from a rumen perspective. So the team have come together, some rumen biologists, etc., have talked about the finding, and there will be further discussion about, can we understand it, or is it just one of those things of simply saying this is our hypothesis of why it's different.
Supplementary question, but if a dairy animal produces most of its methane during the time of its life when it's lactating, is it going to be more important to focus on breeding for that trait than the one we've already achieved?
Yeah, absolutely. So our future research from here will very much focus on lactating animals. So as you state, I mean, the vast majority of methane emissions in the dairy sector come from lactating animals. If it appears from our data set that you need to measure those animals to get the specific genetics of interest, that's where we'll go. And then we'll develop DNA-based selection, so genomic selection, which we apply routinely for other traits. And that will be the approach of selection of higher, or of more obviously better, lower methane producing animals.
Probably got time for one more question. And I say one because I'd like to hear from everybody about it, please. So perhaps we can start at one end and work down. What do you think will drive farmers to use these mitigation tools, assuming that they come at a cost?
Yeah, I kind of hinted it in my last slide that I think demonstrating productivity gains as a result of reducing greenhouse gas emissions is possibly the way to go. And that trial that Sinead talked about may very well help in giving us a quantum number as to just what improvements in productivity will result by reducing methane and perhaps nitrous oxide emissions. At the moment, there's no incentive really for our farmers to take on these other than just public good and goodwill, because they all will have an additional cost associated with them. Would that be passed on the consumer? Don't know. But at the moment, if we were able to show that improved productivity outcome, I think for many farmers that would be sufficient for them to have a look at some of these options.
I kind of feel it's going to be the same answer, but yeah, for us and the conversations we've had, productivity is king. I think with our probiotic technology, we may have the ability to improve the meat and milk quality as well. So I think that's an additional value driver for us.
I think really it's focused on believability, number one, because I think if the farmer believes it's going to work and we can prove it, the other is going to be productivity. I think that's going to drive a lot of the uptake. We're a global aspect too. So I think those carbon markets are developing as well. So to monetize it and show the cost, the return on investment is going to be extremely important. So in that gain, in a lot of the markets, we'll have to also think about the productivity overall.
So I definitely agree with the productivity statement and won't bang on about it. But we've been asking ourselves a number of times throughout the last couple of days, who's going to pay for this stuff, or who bears the cost if we adopt these technologies? And I was wondering the other day, I was talking to someone over a beer, I think it was last night, who bears the cost if we don't? What happens if we don't? And do we get left behind, or do we lose market access, or do we lose the premium of our product or brand New Zealand, or just, I don't know if we spend any time wondering about that, or what happens if we don't bear the cost to do it, what the trade-off is if you just said, I'm not going to be involved in this at all, and other people move forward with it, but I don't.
For me, it's very much about commercial drivers, whether it be from increased productivity, but I think if it's going to be direct for methane or for lower methane production. Brendan showed earlier today, there's a lot of high proportion of our farmers that do adopt. We've seen that with new technologies that were brought to, different businesses have brought to the agri sector. Farmers embrace them when there's commercial benefits. So I don't see this being any different.
Yeah, so obviously the win-win is, you definitely need that. But I think also, and I think a shout out to my other Irish colleague, Brendan, who showed a nice graph of, we had, when you segment, the sort of farmer groups, you've got the early adopters and then you've got the wait and sees. And those guys, they're not against adopting technology, but they need a portfolio of evidence to give them confidence that if they're going to implement them, it's not going to adversely affect their business, one, and ideally make money. So we need to be creating that evidence portfolio for the end user to make sure that they can be confident to take up those mitigation approaches, and it's not going to have unintended consequences. It may hopefully increase their business for all of the aspects that we've been discussing over the 2 days. So for me, it's getting that evidence, creating that certainty that these types of technologies are safe, they're practical, and I think really important, which we're hearing back from sentiment, is independent validation of these technologies. Okay, so farmers need to know who they can trust, that's going to say it's not going to be an issue on your farm. It actually could do good things. So I think increasing that evidence portfolio is going to become really, really important as we move forward in this area.
Yeah, it's interesting to hear several of you say that it's not just about how is the farmer going to benefit, it's them wanting to understand that they're not going to lose by adopting these, because it could feel like a risk or a leap of faith. Yeah, I think there's some kanohiki te kanohi, neighbours, speaking to neighbours, that can happen here. All right, look, we could keep going because there's so many questions, but they're getting quite specific as we go further down the list. So let's enjoy some lunch.